Hello and welcome to my YouTube channel, and today's topic is the classification and characteristics of life, which includes taxonomy and a little bit of history. Biology is all about the study of life, so what makes something to be considered living? And there are 7 characteristics that a living organism has, and they're listed on the screen. But many times when you ask somebody this, they will say that either the organism has to maintain homeostasis, they have to have cells, they have to have DNA. That's not necessarily true, because homeostasis is a result of these characteristics playing out, and hence the organism can maintain homeostasis. And having a cell is also not important, because many organisms don't have cells but they're still living. And DNA does not do anything, it just sits there. It's the proteins that read it in a sense and play out whatever the DNA is asking them to do. And hence, there's some common misconceptions that I've cleared up, and I've given you 7 things that need to be observable in an organism for it to be considered living. And now that we've discussed the characteristics of life, people classify life as well. So you've probably heard like names or specific scientific names of certain organisms, so that's a way of classifying. So let's start off with the history of classifying. And at first it was Aristotle who introduced the first classification system, and it classified plants and animals, and it did it based on size, structure, blood, and where they lived. It was so vague, and hence it wasn't really followed through, and we don't use it currently because it wasn't classified on relatedness that well. Next up, we have Carolus Linnaeus. He made the Binomial Nomenclature, which is a way of classification that we still use today. It's a scientific name, but it has been revised so it can classify more precisely and not just on physical characteristics, because he only classified in physical characteristics. Now moving on, I would also like to mention that classification is very important so that we don't confuse or rediscover animals by accident or something like that, and hence classification becomes something very important in science. Next, we have binomial nomenclature, and that is what Carolus Linnaeus made, and we kind of tweaked it, but we'll talk about the tweaking part later. Basically, the format is having the genus and the species, but it's written in a certain way too, where the genus, the first letter of the genus is capitalized and everything else is lowercase. So for example, for humans we have Homo sapiens. Homo is the genus, sapiens is the species. The H is capitalized because the genus is supposed to be capitalized. Genus and the species are the 2 most specific levels of classification. Now moving on to how we tweaked it to make it more accurate. Before, Linnaeus basically did it on physical characteristics, but now we do it on internal, external characteristics, genetic relationships, which is a biochemical analysis. So basically we look at the genetic code and the order and the specificity of it, the chromosomal composition, which is shape and number of chromosomes, mating behavior, geographical location. So we also take all that into account when using this form of classification. Now we have taxonomy, and Carolus Linnaeus also came up with this, so he's the father of taxonomy. And basically this is a systematic classification of living things into different groups based on physical relationships and genetic relationships. So a taxonomist is a person who studies taxonomy, by the way. And there are different taxonomy levels that he came up with, and they go in this order from most specific to least specific: species, genus, family, order, class, phylum, kingdom, domain. So usually to memorize it, you'd be like domain, kingdom, phylum, class, order, family, genus, species. So that's a good rhyming way to memorize it. And the most specific are the family, genus, and species, and those are the lowest levels. And the species consist of they only consist of 1 organism. And the broadest levels of classification, or the highest levels, are domain, kingdom, and phylum. Prokaryotes versus eukaryotes. Prokaryotes are simple, unicellular, and they have no nucleus or membrane-bound organelles. Instead, they have a cell wall, and instead of having a nucleus, the DNA is held in nucleotides. As for eukaryotes, they're complex, they're multicellular, they have a nucleus and membrane-bound organelles, and the nucleus holds the DNA. So now there's 4 key terms in here that some of y'all may not know. To break that down, we have cell wall versus cell membrane. Eukaryotic cells, and that includes plant and fungi, cells. So the cell wall of plants are made of cellulose, and the cell wall of fungi are made of chitin, and they're fully permeable, meaning they allow all substances to come in and out. They protect things inside the cell, and they hold the shape of the cell because cell walls are rigid and they provide structure. Cell membranes are also known as plasma membranes, and they're found in prokaryotic cells, which are animal cells, and they're made of a phospholipid bilayer with proteins. They're semi-permeable, meaning they only allow certain things to come in and come out. And they help with intercellular transport, which is cell to cell, and they protect the contents inside the cell. So they don't provide structure or shape to the cell like the cell wall does. And the nucleus is in eukaryotic cells, and it's an organelle that holds the DNA in a compact and very protective way, because the nucleus is made of a membrane, and it protects the DNA by allowing only certain things in and certain things out. As for nucleotides, they're in prokaryotic cells, and it's just the area in that cell where the DNA kind of floats around, and the DNA is not very well protected. That's why in prokaryotic cells, like bacteria and stuff like that, there's more chances of mutations because the DNA is not so well protected. Now we have different types of domains, and we have bacteria, archaea, and eukarya. So in bacteria, they're unicellular prokaryotes, which means they lack a nucleus and a cell membrane, and they have a thick cell wall. As for archaea, it's a unicellular prokaryote as well, but the cell wall is different in composition than of the bacteria. It lacks a certain element, and I've listed the name of it, and it contains unusual lipids, and the organisms in this domain live in extreme environments, so they're known as extremophiles. Basically, for example, you have thermophiles or halophiles, and I've listed a few other examples. The domain eukarya has a cell wall and a nucleus, so it's eukaryotic, and it's also one of the most diverse domains you will find, and I have pictures of examples for each one. Now, in eukaryotes, there are 4 kingdoms you should know. So in the eukaryotic domain, there's 4 kingdoms, and they are Protista, Plantae, Fungi, and Animalia. So Protista is basically unicellular. Most of them are ancient eukaryotes, so they're not found in other domains, and they're photosynthetic. Plantae are multicellular, so they're basically plants. And they're photosynthesized, they're autotrophs, meaning they make their own food, they're motile, they lack mobility, and they have a cell wall made of cellulose. Fungi, it's in the name fungus, so fungi is multicellular. It has a cell wall which is made of chitin. Heterotrophs, they grow hyphae, which are long filaments, and they are decomposers. Basically, they release enzymes which decompose the organism, and then they're able to absorb the nutrients, and that's why they're called the decomposers of our ecosystem. And then we have Animalia, which is basically animals. That's basically like us. They're multicellular, heterotrophs. They have to consume others to gain energy or food. They have a cell membrane, and they're gametes at some point in their life. So gametes are basically like a sperm or egg. And now here's a chart of specific characteristics for each, and I listed the domain and the kingdoms that fall into the domain. So in archaea, we have archaea bacteria. In bacteria, we have eubacteria, and in eukarya, we have Protista, fungi, Plantae, and Animalia. So in each one, I listed the cell types and cellularity, cell wall, the trophy, reproduction, and I've listed what each of them means. So cell type, if it's prokaryotic or eukaryotic, is cellularity, is it unicellular or multicellular, or is it both? And then in trophy, that's basically if it's an autotroph or a heterotroph. Autotroph meaning it makes its own food, and heterotroph meaning it needs to eat something else to get its food. And reproduction is whether it's sexual or asexual reproduction. So there we have it, and I tried to organize it the best I could to where you could see which kingdoms fall in which domains, the characteristics of each. Now lastly, to go deep into internal external structures, we have 3 types, which is the first one is vestigial, which is they're part of an organism, however they are of no use right now or anymore, and it's a theory because we can't go back in time to make sure, so it's still a theory, but it's pretty promising, and these structures will disappear over time after use. And for example, we have snake barbs or spurs which jet out in the direction of snake hips, and then we have whales where they have bones where hypothetically the hip bone would be present, and the bone is in the right right shape and place. We have pictures of both. Homologous are same structure or origin but different function, and analogous are similar function but different origin or structure. So that is it for today, and if you have any questions, please comment down below, and I'll be releasing new material on specific topics in biology, and I hope this helps you through high school. Please like and subscribe as I continue to make more content.